Hi everyone, I hope you're all doing well. By the way, good morning, good afternoon, good evening, wherever you are now or for what time you're watching the lecture. By the way, I am from the Ninth Reporter along with my partner, Miss Maricar Maestrado, and I will be discussing about the effects of mutation on our DNA sequence. Okay, so I know that most of you already have a background knowledge about what is mutation all about, but this is just an overview and a recapitulation and also a refreshment for you. Okay, so by the way, we will be discussing effects of mutation on DNA sequence. So basically, mutation is actually a sudden changes from our DNA sequencing. So there are actually two types of mutation, could be somatic mutation and germline mutation. So when we talk about somatic mutation, these are mutation that could be acquired from environmental factors such as chemicals, um, radiations, and also germline mutation could be inherited, and this it occur during cell division. Okay, so by the way, mutation can occur spontaneously or as a result of exposure of various environmental factors such as radiation, chemicals, or viruses, but some mutation may have no effects on the organism, while others may result in genetic disorder or diseases. Okay, so let us now proceed. Okay, so mutation. So these are changes in our DNA sequence. So I already give you the short background or what is mutation all about. So as you can see in these images here, these are the examples of the mutation. So this first image, this is an example of a somatic mutation. So we all know that somatic mutation, these are factors that is acquired from UV rays, X-rays, and harmful chemicals. But this skin cancer is actually acquired from UV rays. And also this other one here, this is a sickle cell anemia. So this is actually common to those person who is or who are anemic. So basically low kaayo ang ilahang blood count or blood cell, and also ang processes or ang production sa ilahang hemoglobin is very baba kaayo. So this skin cancer is actually a common type of somatic mutation from where exposure of the ultraviolet rays coming from the sun. So that is why it is advisable to always wear your sunblock or your jacket or umbrella in order for you to avoid the direct sunlights. Okay, so by the way, class, if you want to have a further if you want to have you know a further examples about this one, you can always download this PowerPoint presentation for your reference and for your note as well. So these are the compare and contrast between somatic mutation and germline mutation. So by the way, the comparison of these two, mutation or somatic mutation, this is actually could be acquired from environmental factors such as chemicals or harmful chemicals, radiation, and viral infections. While germline mutation, this is actually it it occur during cell division, and this could happen due to the exposure of mutagens or viral infections. So by the way, class, not all mutation are harmful. Not all mutations is an exposure from exposure from mutagens or viral infections, but most of the mutation is actually it occurs spontaneously. Okay, so here are some examples of somatic and germline mutation. So as I've said a while ago, somatic mutation um, is an acquired mutation, while germline mutation is a hereditary mutation. Acquired mutation means we could get this one in we can get this one in our environment. So there are environmental factors that can cause mutations such as harmful chemicals, radiations from the sun, and as well as X-rays. Anyways, class, if you are too much exposed from direct sunlight, you might experience skin disease or much worse skin cancer. And also if you are exposed to X-rays, you might experience you know You might acquire cancer cells, because too much exposure from X-ray is very harmful and dangerous. Because X-ray, based from the waves and optics, X-rays have a higher frequency or higher volume of the you know in terms of its wavelength. And also in hereditary mutation, it means that this kind of mutation could be passed on to offspring, or it can be passed from generation to generation. It means it is hereditary or heritable. Okay. If you seek for further examples, you can always refer to this PowerPoint presentation or download in resources from legitimate websites. Okay, so by the way, class, here are some example of the point mutations or somatic mutations. So here are the examples: radiation. So radiation coming from ultraviolet rays, X-rays. And for chemicals, we have smoking and vaping. And for other one is from the foods, from preservatives. So by the way, class, for chemicals, daghan kayo nagaingon like mas nindot pa daw ang vape kontra sa smoking kay mas safe pa daw ang vape. Actually, guys, it's the same thing. They are both dangerous. Just like namili lang ka kung unsa gusto nimo, what type ang gusto nimo, or what I mean, kung unsay way ang gusto nimo mamatay ka, like is it whether organic or vape, is it organic or electronic, because vaping actually is associated from electronic smoking. So they are both dangerous, guys. And also for from foods, we should always be vigilant from the foods we're eating. It's because they might contain benzene, yeah, and other harmful chemicals, just like from the food we're eating outside from We don't even know where they came from. Are they came from the infected animals, or we don't even know. But to be safe, we should always observe what we eat, and most especially from the infectious agents. So here are the two examples: the HPV or human papilloma virus, or the Helicobacter pylori or H. pylori. So we can actually acquire these two types of viruses if we're not actually careful from the water we drink, because most especially this Helicobacter pylori, we can actually get this one from dirty water or contaminated water. So it is advisable to drink, like mineral coming from the water stations, because safe na sila imnon kontra sa, because commonly here in Davao City, like ang ato ang tubig kay murag na-expose siya sa ano, like ang mga hose is na butang siya sa mga kanal. So there are tendencies nga maka-acquire tag Helicobacter pylori. But this is just a rare case. So far wala may reported cases about this one. So this is also the examples of the germline mutation. By the way, this part is for somatic, and for this side is for germline mutations. So germline mutations, these are breast cancers, kani diri sa lang ay sa kuan, diri sa part sa stomach ug diri sa reproductive system. Although cancer is not a direct cause of mutation, but however, they contain cancer cells from the genes, so it could be passed on to offspring and can be passed from generation to generation. But it doesn't mean na if ang imong mama nay cancer tapos ikaw pud magka-cancer, no, dili siya. Like for example, ma-inherit lang nimo siya nga mutation coming from that cell. So taas kayo ang imuhang potential, or taas kayo ang risk na magkaroon kag cancer as well. And same with cystic fibrosis. This is actually a mutation or a disease from our lungs, but commonly na locate jud ni sa lungs. But all in all, or in general, this will ruin your immune system, your reproductive system, so on and so forth. Okay, so I hope this discussion is clear. So let us now proceed to the next slide. Okay, so there are several types of mutation that are classified according to how the DNA molecules is altered. And one type is called point mutation. So it affects single base and most commonly occurs when one base is substituted or replaced by another. And another thing is mutation is also resulted from added or one or more bases known as insertion or deletion or deleting the nucleotide bases. So as you can see here in these two samples here, by the way, class, this is the normal DNA sequencing or the DNA sequence. And this one here is the mutated one. So as you can see that the normal sequence for nucleotide or for the codons is cytosine, thymine, and cytosine. And for this one is guanine, adenine, and guanine. But instead, what happened here, instead of guanine, it was actually inserted with, the mRNA uracil or from the ribonucleic uracil. So therefore, if insertion nga mahitabo diri, therefore there would be an incorrect production of the amino acid which can impact the production of the protein as well. Okay, so just to remember, just to give you some idea that this rDNA sequence is look like a zipper, guys. Na if mamali lang siya og butang sa isa ka bangag or isa ka hole, therefore mali na dayon siya tanan. Okay, so by the way, we will deal with that later. Okay, let's start with the point mutation. So as I've said a while ago, nucleotide base is whether added, deleted, or changed. So as you can see here in this image or illustration, these are the example of substitution, insertion, and deletion. Okay, so mutation that spontaneously occur during DNA replication. But because as I've said a while ago, that not all DNA is being exposed to mutagens or viral infections, but most of the mutation is it occurs spontaneously. But not all mutation are dangerous, but some are beneficial. Or even there are also mutation that is silent or missense. Just but we will deal those ideas later. Okay, so here are types of point mutations. Okay, so number one type is substitutions. So under substitution, there are three types. This is namely nonsense, missense, and silence. So in a class for substitution from the word substitution, just like it was inserted or not inserted but substituted, like napulihan siya daraa. Just see for an example here, instead of guanine, na-replacean siya og adenine from mRNA. Okay, so let's start with the nonsense mutation. So, ginatawag siya nga nonsense, guys. It is because it is considered or categorized as top codons. So, basically, instead nga kuan, instead nga lahi ang iyahang kuan diri ang nucleotide, gipulihan siya og guanine. So, here are the sequences or here are the stop codon, a certain sequence of bases. For example, kining TAG, TAA or TGG. So for example, for the mRNA UAG, so therefore it is a stop codon. So automatically it will terminate all of the production of the kuan, amino acid or protein, or it will terminate everything. So always remember that the stop codon always found at the end of the mRNA. It's because stop na gani siya. It's because makita na siya dito sa pinaka last. Okay, so same goes with the DNA. So for example, TEJ, so automatically stop codon na na siya. Dara na na siya taman or dara na na siya mag-stop. Also, other thing is the second type of substitution is the missense mutation. So like nonsense, missense mutation is actually another type of substitution. So see for an example here, that the codon from the normal DNA sequence is CAT or CAT, cytosine, adenine, and thymine, but instead it was actually replaced or substituted by guanine, which produces the incorrect amino acid that can also affect the production of the protein. Okay, so as you can see right here, that there are actually several causes of substitution or missense. It could be exposure to mutagens or viral infections, or could be um, spontaneous, or it occurs spontaneously. All right, so it doesn't mean that this is a stop codon, but however, it's still functioning. It's still function. Naga-function gyapon na siya, guys, but this is not a stop codon. Nagaproduce lang na siya incorrect amino acid, but this is not a stop codon. And proceeding with the third type of the mutation or insertion, what I mean substitution is the silent mutation. So ginatawag na siya nga silent sa aside sa wala siya factor or dili siya maka-affect sa ato ang body, ginatawag po na siya silent mutation. It's because instead of cytosine and guanine right here, gipulihan na siya og thymine and adenine, and still it produces the same amino acid. Just like it is actually different in terms of nucleotide properties or nucleotide bases, but they are the same in terms of amino acid, but they are the same in properties in terms of codons or nucleotide bases. Okay, so I hope that is clear to everyone. So let us now proceed to the next slide. Okay, so let's proceed with the insertion or deletion. So from the word insertion, it means na insert siya, and deletion it's mean deleting or na-delete ang isa ka nucleotide base. So just see for just look at this example. So here, these are the normal DNA sequence and these are the abnormal one. So instead na mauni siya ang iyahang codons, so since na naman siya na-insert diri therefore kining mga nucleotides diri muisbog na pud na siya muisbog na pud na siya padulong didto. So therefore mausab na pud ang sequencing sa codons. So since mausab siya, mausab pod ang production sa amino acid and which will affect the production of the protein. So that is why this type of mutation is actually dangerous. So later on we will deal about the examples and causes of these mutations. Okay, so here are the three common types of the point mutations. So basically these are a germline mutation. Yeah. So let's start with the cystic fibrosis. This is one of a dangerous disease. It's because gonna destroy ang imuhang baga or ang imuhang lungs. Not just lungs, but your entire nervous system as well as your productive system and as well as your immune system. And for sickle cells anemia, this is actually a common type of point mutation. Like sa mga anemic na mga tao, like ang hemoglobin protein nila in the red blood cell is very kuan, like gamay kaayo ang blood count, the tungod gamay kaayo ang blood count, like mura siya cause sa kuan kanang poor of oxygen. That is why mag-result siya og kuan anemia, like baba kayo ang production sa platelet niya, something like that, but this is really dangerous. Okay, so I hope this is clear. So if you have any further questions or additional idea, you can always download this reference here, or better yet, go to the legitimate website and search for another examples. So here are the summaries. So I already discussed to you about mutation. So mutation, this is actually a sudden changes from our DNA sequence, could be the result of mutagens, exposure to viral infections, and also mutation have two types, could somatic mutation and germline mutations. Okay, so let us now proceed to the second part. So we are now in the asexual prokaryotes as achieving genetic diversity. Okay, so starting with asexual prokaryotes such as bacteria. Okay, so we all know that prokaryote, these are the multicellular cells, if I'm not mistaken. By the way, just for a moment, class. Okay, actually, I am wrong. So prokaryotes is actually a, you know, it has a cell, but there's no nucleus in it. So unlike eukaryotic cells, so examples of prokaryotes are bacteria. They do not produce through sexual reproduction, which typically leads to genetic diversity through the mixing or shuffling of the genes or shuffling of the genes. So these organisms are able to achieve genetic diversity in general ways. So here are the ways in order for this organism to adapt. on its environment or to adapt or to achieve the genetic diversity. So we all know that a beneficial mutation can provide an advantage to the organism by improving its ability to survive and reproduce in its environment. So, see for an instance, if a mutation can confer resistance to an antibiotic, can allow the prokaryotic to survive in the presence of antibiotic. So, there is actually a contribution of the mutation from this type of organism. It's because when they are mutated, they change their characteristic. So instead nga mamatay na sila daraa, but instead they can respond to stimuli, maka-adapt sila og bag-ong environment which is beneficial to them, just like for example ma-involve sila sa ingan ng environment nga murag dili nila nga andan, but because of this mutation ma-adapt nila ang ilahang environment which is beneficial pud sa ila because it will change their characteristic as an organism. Okay, so by the way, there are classification of this mutation. Number one is the beneficial mutation, and the other one is harmful mutation. So the other thing is harmful mutation can have the deleterious effect on the organism. So such as reducing its fitness or causing diseases. For example, that disrupted. I'm so sorry. So, for example, a mutation that disrupt an essential, metabolic pathway is an a prokaryote they could prevent the organism from synthesizing an important nutrient, leading to the growth of impairment of even death. So, like harmful mutations, like makakause siya death sa isa ka organism, it's because like harmful gani no, na di ay harmful nga dili makakuan. So, it is understood the beneficial and harmful mutation. So other thing is the other way that can prokaryote can adapt the genetic diversity is the horizontal horizontal gene transfer. So for the horizontal gene transfer, there are three mechanisms right here, which is the transformation, transduction, as well as conjugation. So in horizontal gene transfer, actually, guys, this is um, it transfer from genetic materials between different organisms that are not parent and offspring. So, in other words, you should always remember that it involves the movement of genetic materials between these organisms that are not related to descent, such as between bacteria or between bacteria and eukaryotic organism. So, by the way, in other discussion, the horizontal gene transfer can occur through several mechanisms, including transformation, transduction, and conjugation. So, these are the mechanisms right here. And also in other discussion from researchers, in transduction, um, for a moment, guys. So I'm so sorry. For in transformation, bacteria take a free DNA from the environment and it incorporate it into their genome. So in other explanation in transduction, bacteria viruses called bacteriophage or bacteriophage transfer genetic. It transfer genetic material between bacteria. Okay, so later on we will deal about the transgenic organism because this factor will apply on that concept. Okay, so lastly in conjugation, it explains that bacteria exchange DNA through a physical bridge-like structure called pilose. Okay, so just to sum it up, that horizontal gene transfer can have important evolutionary consequences as it allows organisms to acquire new traits and adapt the changing environment more quickly and through traditional mechanism of genetic variation and also the natural selection. Just see for instance, bacteria can acquire resistance to antibiotic through HGT, allowing them to survive and thrive in presence of this drug. Just like magkaroon sila og genetic diversity or resistant mechanism instead nga mamatay na sila ana nga certain component, but because of this horizontal gene transfer maka-adapt sila og resistance like resistant components. All right, so let us now proceed with the replication error. So even during a sexual production, error can occur during DNA replication that lead to the genetic diversity. This error can result in the production of new alleles or variation of gene expression. So just to explain this one, the replication error refers to a mistake that can occur during DNA replication. So nag-occur na siya during DNA replication just like na error during the division or sequencing of the DNA, which is the process by which cell make copies of their DNA prior to cell division. And also DNA replication is a highly accurate process. Yes, but errors can occur due to the various factors, including DNA damage, exposure to mutagenic agents, such as, radiations or harmful chemicals, or viral infections, and make I'm so sorry, and mistake made by the enzymes involved in the DNA replication. Okay, so there are actually mechanisms here in order to minimize this replication error. So number one is to minimize the impact of replication error. Cell must have several mechanisms for error correlation and proofreading during DNA replication. And this includes specialized enzyme that can detect and repair mismatch or error in the DNA sequence. Okay, and as well as checkpoint mechanism that can delay or halt cell division until error are corrected. Okay, but however, these mechanisms are not foolproof. Foolproof or it means a replication error can still occur in a low rate in a normal cell. So though naa siya yung mga factor or mechanism nga i-prevent ni siya, but napod mga factors nga dili jud nato malipayan nga malikayan nga mahitabo gud siya. Mahitabo jud siya. Okay, I'm sorry for that. So let us now proceed to the other slide. So we will be talking or dealing with the modern application of microbial genetics. So as you can see right here, scientists using biotechnology to improve the human life or in order to benefit the humankind. So up until this generation, biotechnology is still improving and still prevailing. So this is also important for us nga para dili ta mawad-an og mga supplies, or in order also for us to domesticate plants and animals in order for our, in terms of our survival as well. So domestication, this is actually common. So if you can see the video from the YouTube based from the history of domestication, this is actually a domestication of plants and animals, and up until now, it is common. And this biotechnology also apply to transgenic organism, but later on we will be dealing that one. Okay. By the way, yes, it is actually correct that biotechnology is a broad field that encompasses the wide range of applications of living system to benefit humankind, including you know, including the use of microorganisms, plants and animals in agriculture, medicine, and industry. And the domestication of plants and animals through farming and breeding practices can be considered a form of biotechnology because it involved intentional manipulation of living organisms to achieve desirable trait of human use. So there are actually um, not factors, but positive factors about using biotechnologies. Just like, for example, the production of insulin. They actually getting the sample from DNA or for the insulin gene from the human, and as well as plasmid from the bacterium. They actually merge that one or combine. So what can we call that one is the recombinant process, from where the insulin gene of a human and a plasmid from bacterium, they actually combine that one to produce an insulin. And we should know, it is actually common that bacterium is also rich in production of insulin. So they actually get the sample from the donor, which is the insulin gene from the human, and also the recipient or the host coming from the bacterium. They actually merge that one called recombinant, and later on they sell the samples, and a few months pag-check nila is nagkadaghan ang production sa bacterium that contain insulin. So we all know that insulin is actually one of the biopharma, like mura siyang medicine nga i-inject sa isa ka tawo nga naay diabetes or diabetic person. So ginagamit na siya in order to lessen or to to decrease the risk o para ma-decrease ang risk na ma-trigger ang isa ka disease or sakit. So that is the major contribution of the biotechnology into human life. Okay, so I already discussed to you. So these are other example of biotechnology or cloning process. So molecular cloning. So as you can see in this example right here, that they're actually using plasmid from bacterium and insulin gene from the human. They actually combine that one called recombinant or recombination of DNA, which will later on they will produce a an insulin or human insulin. So kuan gyud siya kanang beneficial jud siya sa mga tao, like makatabang siya sa mga naay sakit, or this is also helpful and beneficial to farming, most especially sa mga crops or sa mga tanom good like mais, sa mga humay, like nagakuha sila og toxin from other organism o gina-inject nila sa mga particular plants so that katong mga parasite nga mukaon sa mga plants og mga crops is mamatay sila. It's because those plant already contain toxin from other organism, which is also beneficial from everyone. And also those plants that inherited toxins, they are also they are also classified for, or they are also classified as MGO or GMO. I'm so sorry. I mean GMO or genetically modified organism. It's because nahaluan na siya og mga process, like nahaluan na siya og process, like ma-injectan siya og mga chemicals, like something like that. Okay. So I hope the discussion and example is clear to you. So let us now move on to the next slide. Okay, so let us now proceed to the molecular cloning using conjugation and transduction. Okay. So by the way, molecular cloning is this is actually a technique used by the scientists from the Freebius research. So it used in molecular biology to create multiple copies of a specific DNA sequence or gene of interest. Just like see of this example here. So as you can see in this example here, that ginagamit ni siya kining molecular cloning para magpadaghan og cell, ay, so sorry, magpadaghan og cell or DNA. And also, the process involved the inserting the DNA sequence into a vector, which is a small piece of the DNA, can replicate independently of a chromosomal DNA in a host cell. So in other example here, the vector is then introduced into a host cell, such as bacterium or yeast, which replicate the vector and production many copies of the DNA sequence. So actually guys, if mag-rely lang ta sa human insulin, so dili na siya mag-produce. Kung dili na siya mag-produce guys, taman lang na siya di as the if a person gets older, ang iyahang human insulin is whether magka-decrease na siya. So later on the scientists found out that this bacterium is actually producing an insulin. So na sila experiment nga ginabuhat using biotechnology. So ang tawag atong bacterium nga na-involved sa experiment is called a transgenic organism na kay because na undergo naman siya sa isa ka experiment. Just like for example, we all know that bacterium is actually mag-generate jud na siya, magkadaghan na siya. So what they did here is nagkuha sila chromosome or insulin gene from the human and also a plasmid coming from the bacterium. So gina-merge na nila didto. But then, since successful man ang research, katong bacterium is nag-disseminate siya or like nagdadagan siya. So nakita nila dito nga nagadagan jud ang production sa sa bacterium that contains human insulin. Kinabutangan naman na siya gene sample or insulin gene coming from the human sample. Okay, so I hope you understand well. So please bear with me, guys, because I am not perfect. Okay, so yeah, magsinabdanay rata diri And if you need more examples, I believe that you're media literate. You can actually search from the legitimate website if you need more examples, or if you don't understand my discussion, then you're free to, you know, check for another resources yes, you can always do that. Okay. So additionally, just to sum it up, molecular cloning is a technique used in molecular biology to create multiple copies of the specific DNA sequence of a gene of interest. So I already mentioned about the molecular cloning making insulin for human. So yeah, so I hope the discussion is clear to you, and let us now proceed to the next slide. So we will be dealing the introducing recombinant molecule into eukaryotic host. Okay. By the way, guys, introducing recombinant molecules into eukaryotic host involves the transfer of foreign DNA in the cell of a multicellular organism such as plants, animals, and in another examples, there are also processes typically involve using vector, which are vehicle that can carry foreign DNA into a host cell. Okay, ang purpose po ani is if na insufficiency of production or productivity sa isa ka organism, ginagamit po nato ning biotechnology to like ginagamit po nato ning molecular cloning para mag-produce og other mag-produce of other organism just like sa mga hayop, sa mga plants. So for example, nagka-decrease na ang for example, this particular plant is actually decreasing now. So what unsay ginabuhat sa mga scientist is ginapangitaan nila paraan nga makasurvive gihapon na siya nga plant which is nagahapit na mag or endangered na kaayo. For example, gina-transfer o ginaputol nila ang sample then gina-insert dito sa pikas nga plant. So this process also considered molecular cloning. Dili lang siya pinaagi sa inject-inject but pinaagi po siya sa in terms of farming. Okay, so one common method of introducing recombinant molecules in eukaryotic host in transfection, which involves the use of chemical or physical agent to transfer the DNA into the cells. So ginagamitan na jud ni siya og mga chemicals. So in order to transfer or clone the DNA sequence from other organism. So see for instance here that it involves the use of lipid-based molecules that can bend the DNA and facilitate its, the uptake by the cell. And also in yeah, we will now proceed to the electroporation. So electroporation is actually in other hand, it involves the application of the electrical pulse to the cell to create temporary pores in the cell membrane, allowing the DNA to enter the cell. So as you can see in this picture, guys or class, that this is the plasmid. So what is happening right here, mag-create na siya temporary pore in order for this recombinant DNA to enter inside the plasmid membrane or the plasma membrane. So if nana na siya dira sa sulod, dito na mag-generate tanan ang dito na siya mag-regenerate. Kay nakasulod naman na siya just like kuan mura na siya egg o kini ang sperm like nakasulod na siya. Ang purpose of this electroporation is it will make a temporary hole in order for this recombinant DNA to enter inside the plasma or the plasma membrane. Okay, so another thing is okay, by the way, I almost forgot. There are actually process of electroporation involves applying the electrical field to the cell to create temporary pores. So I already mentioned a while ago that it allows the foreign DNA or other molecules to enter the cell. Okay, so I hope my examples and discussion is clear to you guys. But again, if you need further examples and explanation, you can always search for legitimate website, and you can always do that. So let us now proceed to the microinjection. So this is actually the example of microinjection. Just see for instance, yeah, this example is actually a egg and also this is a sperm. What happened here is ginagamitan nila og kuan gi-inject ra nila ang egg inside the cell. So dako pud siya nga help. So up until this generation ay no, this new generation kay naga-evolve naman jud ang mga scientific processes. So ang uban kay magkuha lang sila egg sa uban nga donor DNA insert dito, or ang nahitabo na karon nga generation is ginapalit lang ang baby coming from the host. Like mga ilaha, like ang sperm is coming from the other donor, then i-inject lang dito without having a sexual intercourse. But in other example here, an alternative method of transfection is called micro injection. Because eukaryotic cell are typically larger than those of prokaryotics, DNA fragment can sometimes be directly injected into the cytoplasm use the glass. So the process of microinjection involve using fine glass needle that's smaller than the cell itself to deliver the foreign DNA or other molecules directly to the cytoplasm or the nucleus of the cell. So the cell are typically suspended in buffer solution, and the needle is inserted in the cell under the microscope. So actually, guys, there's a lot of video in the YouTube, and you can actually the you know watch the short clip about this one. Like common na kaayo siya karon pero super mahal lang siya, like kakita ka tong famous nga Filipino artist, like nagkuha lang siya og samples sa iyang sperm, then gi-inject niya dito sa recipient or from the host. So later on kung ma-develop og mugawas na tong bata, dito na siya niya kuhaon without having sexual intercourse. It's because bayot man gud ang kuan recipient. What I mean, bayot man gud ang donor. So dili na niya gusto makipag-intercourse sa babae because dili niya gusto. Pero gusto niya magkaroon og baby. So what happened here is gikuha niya ang iyang sperm ug gi-inject niya dito sa lain. So that is the purpose of microinjection. So para malesen na, ay para nakatabang pud na siya sa atoa. Okay? So I hope nasabtan to ninyo. Okay, so gene gun. So from the word gun, it means barilon ka. Char. Dili bitaw. So gene gun is ginjan is also common as a biolistic particle delivery system. Like it is a technique used in molecular biology and biotechnology to introduce foreign DNA or other molecules into cell using high velocity micro projectiles. And also the process for this one, for the gene gun, is it involves coating tiny gold or tungsten particles, which the parent DNA or other molecules, and the shooting them into a target cells using high pressure helium gas. Like ginabaril lang na siya, like ginapusil nila tong targeted cell para musulod tong kuan cell coming from the gene gun or the gene. And also the particles are accelerated to high velocity by the gas and are driven into the cell. Okay, so mauni siya ang mga processes where they deliver the foreign DNA or other molecules. Okay, so let us now proceed to the next sample here, which is the viral vectors. Excuse me. Okay, so this image is an example of a viral vectors. So as I have mentioned a while ago, nakatong magbuhat og temporary hole or pore in order for the recombinant gene to enter inside the um you know plasm, basta membrane, something membrane, like cell membrane, I'm so sorry. Okay, so by the way, viral vectors are genetically modified viruses. So GM kuan gyapon na siya, like genetically modified viruses na siya gi-apply nga mga chemicals na, like dili na siya pure, na siya nahagol nga mga substances due to the process of biotechnology. Okay, so these are viruses are used in molecular biology and biotechnology to deliver foreign DNA or other molecules into the cell. Okay, so viral vectors, mo ni siya ang process. Like, ang gamit lang ani is virus ni siya, like genetically modified virus, and virus enter the cell through membrane. Okay, so the process for this one is it involves removing the viral genes responsible for causing disease. So what happened here, ginakuha nila tong mga viral genes good, katong mga genes nga naa sa sulod which cause diseases, ginatanggal to nila o ginapulihan og like genetically modified viruses. Okay, so the modified viruses is then introduced into the target cell where it delivers the foreign DNA and other molecules. So in other instances, guys, the viral vector can be designed to target the specific cells or tissue, making it a highly efficient and targeted method for introducing foreign DNA inside the molecule or inside the molecules into the cells. Okay, so I hope nasabtan to ninyo tanan, guys. But again, if you need clarification or further examples, you can always search to legitimate websites if you want another or additional examples. Kung wala ko ninyo nasabtan pag research mo dito kay I believe media literate naman kaya mo. So please bear with me, guys. Thank you. So let us now proceed to the last topic here, which is the whole genome method and pharma like naglisod ko na ni pharmaceutical application for genetic engineering. So for genetic engineering, these are the processes here. So for the involved of DNA, ginagamitan siya genomic process or mechanism. And for mRNA, ginagamitan siya transcript tra- transcriptomics. And in terms of the proteins or protein array, ang tawag sa process is proteomics. Ay, yes, proteomics. Let's dive deep sa description. Okay, so let's start with genomics, guys. So by the way, genomics, this is actually based from previous researches. This is the study of the structure, function, and as well as evolution of genomes, which are the complete sets of genetic materials, including genes and non-coding DNA, within the organism or a population. So mauna siya ang iyahang function based sa conducted study from researchers. So genomics is an interdisciplinary field that involves the use of molecular biology, genetics, and uh, compu- computational biology as well, and also in biopharmatic to analyze the or to interpret the vast amount of genomic data. Yes, guys. Okay. So in other thing, naapud mga field that that it revolutionized our understanding about genetic basis of biological processes. So the field of genomic has revolutionized our understanding, by the way. So including development, the growth, and as well as disease. Okay, so by this sequencing and analyzing the genomes of different organisms, it will be able the researchers to identify the genes and other functional elements, and also their interaction and regulation based from the previous research. Okay, so in other explanation here, it leads to the discovery of new genes and pathway involved the various diseases as well as new targets of drug development and personalized medicine. So I know, guys, that this explanation is really broad and really complex to comprehend, but if you're having trouble to understand the discussion, you can actually free to research from the legitimate website for additional ideas, because 90% of this reference is coming from the instructional manual and another percent is coming from the legitimate source from Google. Okay. Okay, so I hope everything is clear to you. So we will now move to the next example here. Okay, so let us now proceed to the transcriptomics. All right. So just to discuss this one, transcriptomics is the study of the transcriptome, which is complete set of RNA transcripts produced by the genome of an organism or a cell at particular time or under specific condition. And also transcriptomics or transcriptomics is a rapidly growing field that has revolutionized our understanding of gene expression and regulation, and as well as the function of non-coding RNA. Okay, so by the way, just to explain what is transcriptome all about, it includes different type of RNA molecules, such as mRNA or messenger RNA, which carry the genetic information from the DNA to the ribosome of protein synthesis. And in other way, it also involved the non-coding RNA, which does not encode protein but have important regulatory function in gene expression. So even though dili siya coding RNA, naa po siya important factor sa ato ang DNA sequencing. And this factor is analyzing the transcriptome. Okay, so by the way, based from the researchers, they will be able to identify the different expressed genes on alternative, sorry, alternatives, alternatives placing events, and post-transcriptional modification and study the rules in various biological process and diseases. So, mo na siya ang importance of transcriptomics. And also, let us now proceed to the metagenomics. So, metagenomics, when genomics and transcriptomics are applied to intermicrobial communities, we use term metagenomics and metatranscriptomics uh, respectively. All right. So, by the way, class, met, metagenomics is a study of the genomic content of a mixed community of organism. So, literally, this is a mixed community of an organism such as those found in environmental samples, such as soil, water, and human gut. Unlike genomics, they are actually focusing on the genome of a single organism, while metagenomic involves the sequencing of analyzing all of the genetic materials in a sample. Just like gina-combine nila ang community or gina-combine nila ang mga organism into a community. All right? So, while metatranscriptomics, yeah. On the other hand, this study is the study of the transcriptome of a microbial community. So in other words, this include all RNA transcription produced by the community membrane. Okay, so like metagenomics, metatranscriptomics provide a snapshot of the functional activities of a microbial community of a popular time and under specific condition. So in other thing, it analyzes the metatranscriptome, or the researchers can identify the genes that are actively expressed and metabolic pathway that are active and the regulatory mechanism that control gene expression in the community. Again, guys, I know that the expression or the explanation is very broad and complex to understand, but again, there are other resources from a legitimate website. You can always go there and search for another examples if you're having trouble understanding the discussion. By the way, guys, let's proceed to the other example here for the toxico. Ay, sorry, Texo, toxicogenomics. Sorry. Basa na kog tarong ha. Toxico, Toxicogenomics. Yes. So this study, this is actually the interaction between environmental toxic and genome. So environmental or interaction between environmental toxic and the genome. So gitawag siyang atay man nga word oy, toxicogenomics. It's because gi-combine ang genome ug ang toxic and genome. So toxicogenomics. Sorry. So, by the way, class, typically toxigenomics typically involves the analysis of gene expression, profile in response to exposure of the toxin or the chemicals. So, this often used in high methods such as microarrays or RNA sequencing by comparing the gene expression patterns in exposed and unexposed cell of organisms. So this toxicogenomics research can identify different expressed genes, pathways, and as well as network that are associated with the toxicity. Like ginakuha nila ang mga toxic sa isa ka gene. Okay guys, if wala jud mo kasabot, you are free to search the other samples from legitimate websites. Or you better yet, you can watch videos if you're having trouble understanding the topic. Again, I understand this is really complex, but again, let's try our best to gather some resources. I know you're all smart and intelligent, so there's no problem at all. Okay, so let's proceed to the the other part, which is the proteinomics. It called proteinomics because it involved proteins. Okay, this is the study. Is an extension of genomics that allow allow scientists to study the entire complement of protein in the organism called the proteome. Ambot of unsay basa ani proteome ba ni? So even though all cell of a multicellular organism have the same set of genes. By the way, guys, in other examples here is it relies on a variety of high throughput techniques, including mass spectrometry, protein microarrays, and protein-protein interaction arrays. By the way, this method enabled the identification of the qualification of thousands of protein spontaneously or simultaneously providing a comprehensive view of the proteome and to change the response of different condition of stimuli. Okay, so mauna na siya guys. Again, proteomic, it involves protein. Okay, it relies on a variety of techniques, including mass spectrometry, protein microarrays, and protein-to-protein interaction arrays. And also, just to add this to your idea, this is the study of entire complement of protein expressed by a cell, tissue, or organism. So, in other thing, it involves the large-scale analysis of protein or protein structure, its function, and interaction with the goal to understanding the role of protein in biological processes and disease. Okay, so let us now proceed to the last part here. So, recombinant DNA technology and pharmaceutical products or production. I'm sorry. So, recombinant DNA technology, also known as the genetic engineering, has revolutionized the production of the biopharma. So, it also involved the use of genetic technique to manipulate DNA sequences and create new combination of genes and can express in living cell to produce therapeutic proteins, antibodies, and other biologics. So, na-explain na ni siya ganiha based sa transgenic organism or molecular cloning, just like production of the insulin. Ginagamit ni siya nga recombinant or genetic engineering to produce a like sa medicine, in terms of crops, in terms of farming, in terms of domestication of animals, something like that, like applicable jud siya sa human being, although there are negative aspect, but we will not focus on that one. So recombinant DNA, we already discussed the recombination. So recombinant, this will help to develop technology in pharma or in biopharma production, just like atong example ganiha good nga production of insulin, like insulin gene from human and plasmid sample from the bacterium. Like, merge sila to produce a human insulin. Okay, okay, everyone. So that would be my discussion, and I hope you learned something or learned a lot. But if not, you're free to to research from legitimate websites since dako naman mo, you can do everything. Okay, thank you so much and God bless everyone. That would be all. Thank you. And by the way, if you need reference for your notes, you can always download this PowerPoint presentation. Thank you everyone and God bless and have a great day.